Well, let's get started. Good afternoon, and welcome to the Cancer Center Symposium series. And I'm Bifeng Gao, and the director of the Genomics Shared Resource at the Cancer Center. And I'd like to take this opportunity to thank all our supporters and who utilize our core for your genomic research. And we're also immensely thankful to the Cancer Center for providing us this opportunity to showcase how our shared resource support cancer research for our members. Today, I'm delighted to introduce our speaker, Dr. Sky Lee. And Dr. Lee earned his bachelor degree from the University of Michigan, Ann Arbor, and completed his PhD in structural biology and biochemistry right here at the CU Anschutz, under the mentorship of Dr. Gongyi Zhang. Following that, and he pursued his postdoc research in immunology at the National Jewish Health, and working with Dr. Pippa Merrick and Dr. John Kipler. Throughout his research career, Dr. Lee has made significant contributions, and with his findings being published in numerous journals such as eLife, PNS, and Cell Research, and many others. So Dr. Lee joined the Genomics Shared Resource and at the end of 2021, and has since served as Assistant Director of the Genomics Shared Resource for nearly 2 years. So today, Dr. Lee will present on the topic, applying advanced genomic technologies in cancer research. And please join me. Welcome Dr. Lee to the podium.
All right, thank you very much for that wonderful introduction, and thank you very much to Cancer Center for inviting me today to speak. Yes, so my name is Sky Lee. I'm the Assistant Director at the Genomics Shared Resource, and it is my great honor and pleasure to talk to you today about applying advanced genomics technologies in cancer research. And we have a lot of grounds to cover, so without further ado, let's begin. Some learning objectives. This is some of the items that I'm hoping that we can cover today. Number one, determine when bulk RNA-seq, single-cell RNA-seq, or spatial transcriptomics is applicable or suitable. Number two, understand the underlying mechanism for various advanced genomic technologies. Number three, what are some novel and representative cancer-related discoveries determined using advanced genomics technologies? And number four, who are some of the CU Anschutz investigators who are generating these results to address cancer research, so that hopefully that opens up for potential for establishing collaborations or seeking some guidance and whatnot. And today, my talk is a little bit different than the ones I usually give, which really focuses on the technology and when that might be suitable and some of the new technologies we have available at Genomics Shared Resource. But rather, we're going to be talking a lot about the research side of things and some of the experiments that we've conducted and the outcome of these experiments. And specifically, what's special about these experiments is that the Genomics Shared Resource has contributed either directly by performing the experiments in its entirety, that is to say from samples to FASTQ, or indirectly by performing next generation sequencing for libraries prepared by the group, that is to say from libraries to FASTQ. And the important part is that all showcased experiments can in fact be performed by the GSR in its entirety. And here are some of the ones that we'll be talking about: methylation array, bulk RNA-seq, next generation sequencing, single-cell RNA-seq, and spatial transcriptomics. And again, all showcased experiments are from CU Anschutz researchers and have significantly advanced cancer research. And as far as that last point, I'll let you be the judge of all that. But I don't know, it was really gratifying for me to at least look at some of this research, and at least the impression I got is like, yeah, it's really moving the field forward. So the first technology we'll talk about is methylation array. Now, it's kind of simple, but I'm not going to assume that everybody knows what this is. So let's kind of go over this a little bit here. So what is DNA methylation array? Well, that starts with the process of DNA methylation, right? Where DNA methylation is a biological process by which methyl groups are added to DNA molecules. So in the mammalian genome, DNA methylation involves the transfer of a methyl group onto cytosine to form 5-methylcytosine. And DNA methylation can influence gene expression by affecting the interaction with DNA of both chromatin proteins and specific transcription factors, where, for example, when located in a gene promoter, DNA methylation typically represses transcription. DNA methylation is essential for silencing retroviral elements, regulating tissue-specific gene expression, genomic imprinting, and X chromosome inactivation. And essentially, DNA methylation in different genomic regions may exert different influence on gene activities based on the underlying genetic sequence. And as far as that last point goes, underlying genetic sequence, that's going to be really important because the magic ingredient in all of this is the array itself, which you kind of see in the lower right-hand corner. That's what a methylation array looks like. And this allows you to actually screen up to 850,000 biologically significant regions of the human methylome. And again, I'll kind of go over that a little bit in the next section or in a little bit here. But what does it all start with? Well, it starts with you providing us with genomic DNA as the starting material. And essentially, this genomic DNA undergoes a process called bisulfite conversion. And really, there's two different outcomes depending on the methylation status of the DNA. So let's take the easy example where, let's say the DNA is unmethylated, right? Which you see in the kind of the blue rectangle. The cytosine undergoes this chemical process where it gets converted into a uracil, right? However, if the cytosine is methylated, so 5-methylcytosine, it is actually unaffected by the bisulfite. So bisulfite converts cytosine residues to uracil, but leaves 5-methylcytosine residues unaffected. So here's again an example right here. So here you get C, G, that C is methylated, and after bisulfite conversion, it stays the C as a C, but here it's unmethylated. So after bisulfite conversion, turns to a U. After that, it undergoes whole genome amplification and enzymatic fragmentation. But during that amplification process, that U is interpreted as T, which is kind of obvious, right? The U is the analog for T in RNA and DNA. So and so then that amplified, fragmented, and then finally hybridized to the array itself. And if we zoom into the array, like I said, there's 850,000 different targets and biologically significant regions, and that's essentially determined by these sequences, right? So these sequences are already predetermined, they're already known, and that's essentially, apparently biologically significant. And depending on what the methylation status of your genomic DNA was, you'll have two different outcomes. So let's say it was methylated over here, so that C remains a C, right? The bisulfite just had no impact on that C. So here, we're gonna, flow some fluorescently conjugated nucleotides. So in this case, the G is covalently conjugated to a green fluorophore. So it binds, and when we put it into the iScan the array scanner, it'll fluoresce green. And okay, so now we know that this locus was unmethylated. In contrast, recall that C was converted to a U, and now it's a T. And here, it's not going to be the G that's binding, but rather an A, right? And the A is conjugated to a red fluorophore. And likewise, when we scan it, it's going to show up as red. And, sometimes you have, some that are semi-methylated. So in that case, you'll have, the red and the green fluorescing simultaneously. But essentially, this is the way in which we determine methylation status of your starting material. So that's first thing. The second thing we'll talk about is bulk RNA-seq. Now, I'm assuming that many of you already know bulk RNA-seq, but maybe some of you don't. So let's still go over it. And by the way, when we talk about some more complicated things like single-cell RNA-seq and spatial transcriptomics, it'll kind of build on this anyway. So for those of you who know, it'll be a nice refresher. And for those of you who don't know, good, like you learned something, right? So what is bulk RNA-seq? Well, that all starts with RNA that is extracted from pooled cell population, tissue sections, or biopsies. And there you go. Like, let's say you have a mouse, you extract some tissue out of it, or you're doing some cell culture. Essentially, you take that and you homogenize it and lyse it using guanidinium phenol, or as we better know it to be trizol, right? Afterwards, you're going to have it lysed, and you have everything in there, but hopefully you're working under a hood. So you put some chloroform in there. It'll undergo phase separation, where in the aqueous portion, you're going to have the RNA, whereas everything else is just down there. I think the DNA is actually in that portion right there. But anyway, then you take that aqueous portion, you put it in a separate tube. I use isopropanol or something else, just kind of ethanol, whatever, to precipitate it. You spin it down. You got the RNA pellet down there. Take out everything else, resuspend it in water, and there you go. Now you have essentially total RNA in a tube that will be used for bulk RNA-seq. And what does it do? Well, bulk RNA-seq measures the average expression level of individual genes across hundreds to millions of input cells. And it's useful to get a global idea of gene expression differences between samples. And of course, that's really important, right? Hopefully you design your experiments where you have a robust control so that you can actually, compare your data, like a treatment versus control, so that this data can essentially be interpretable. But let's say you got the RNA. Well, essentially, you give us the RNA, and what do? Well, there's many different kinds of bulk RNA-seq, but the most popular one that we do is called polyA-selected mRNA-seq. So this is going to specifically look at the transcriptome only, the mRNA only. And that all starts here, where, as you can see, you have this tube, you have all the RNA in there. And really, When we say total RNA, like what, 95% or more, that's going to be ribosomal RNA, and only a very small fraction of it is going to be mRNA. And not only that, you got long non-coding RNAs, tRNAs, just all sorts of RNAs in there. And we want to actually select for the mRNA. So how do we do that? Well, the first thing that we do is add some magnetic beads that have poly-DT oligos conjugated to them. And those poly-DTs are going to hybridize to the poly-A tail of the mRNA, right? So far, so good. And then we use a magnet to pull down those magnetic beads and everything that's just sort of left over in the supernatant, we get rid of it. And essentially, we elute the mRNA from the beads, and what we're left with is just the nice clean tube of mRNA only. And there it is, poly-A selected for mRNA, right? And after that, now it's ready to undergo a process called library prep or library preparation. Again, this might be a term that many of you have heard, like a library prep. Well, essentially, this is it. Like, this is what library prep looks like. You have the mRNA, there you go at the top, that we're starting with, and undergoes fragmentation. And by the way, I'll say this too. So why do we have to fragment it? Well, I'm going to be talking about next generation sequencing in the next section, but another kind of catch-all term for next-gen sequencing is short read sequencing, because in the vast majority of cases, I know there's exceptions, but in the vast majority of cases, we can only read 300 bases of the insert. So if you have like 1,000 base pair insert, I mean, yeah, we might read like 150 from one end, 150 from the other end, but that middle 700 is going to be missing. So, we have to fragment it to about like 200, 300 base pairs so that we can actually make it compatible with next-gen sequencing. So there's fragmentation happening. We have some random hexamers in order to prime for cDNA synthesis and undergoing reverse transcription. So there you go. It's coming in from this side to make a cDNA, and then we'll come from the other side to make the second strand. So now we have a double-stranded cDNA derived from the mRNA, right? Afterwards, it undergoes a process called indexing and adapter ligation. Again, this process is just ubiquitous across all next-gen sequencing. This is the process in which we make the insert actually compatible with next-gen sequencing. So it entails two different points. Number one is the adapter itself, which is denoted P7 and P5. So that's going to be really important in the next section, but the P5, P7 adapter. And then you got the I7 and I5 index. So that is going to be sort of like your sample-specific ID barcode, right? So for example, if we have two customers that have this identical I7, I5 index, like we wouldn't be able to determine like which sample belongs to who. So that specific I7, I5 index becomes really important for next-gen sequencing. But nonetheless, you do all that, you ligate those, and then here you go. You got the final library right there. And afterwards, we do next-gen sequencing. So what is next-gen sequencing? I mean, it's not like it's a noun that I can point to and say, oh, there's next-gen sequencing right there. No, it's a process, and the process starts with a flow cell. And what you're seeing here is kind of the most common flow cell that we use at the Genomics Core. This is the largest one, and this is actually how we managed to afford to give it a cheap sequencing here locally. But if you actually zoom into the flow cell, so let's kind of take a closer look at what's going on there. The flow cell is coated with oligos, okay? So these oligos are covalently bound to the surface of the flow cell, and these oligos are going to exhibit two different sequences that are going to be complementary to the P7 and the P5. So that's why we had to do that library prep early on, where we're ligating those adapters. And so there we go. Now it's annealing right there. It'll undergo DNA extension in order to make a reverse strand right there. And now that particular strand is covalently bound to the flow cell, right? And from there, this one's gonna like bend over like that. And then it's gonna come the other way to make the complementary strand. And now consequently, we have both the forward and the reverse strand on the flow cell. Well, that's not all, because it's going to go through that process over and over and over again while it's just kind of bending over and doing so. It just kind of look like a bridge. So we call this process bridge amplification. And essentially what it does is it generates a cluster. So you might have heard a term before, to cluster. So these clusters, there's many of them. And each of these clusters are all going to be derived from one molecule of that library, right? So essentially the sequences here are all going to be the same in this particular cluster. And that's going to be really important, because the actual readout for the next-gen sequencer, believe it or not, is fluorescence microscopy. So you have these 4 nucleotides that are fluorescently conjugated. So let's take a look here. I think we have a G that's conjugated to a blue fluorophore, a C to a green fluorophore, a A to a yellow fluorophore, and a T to a red fluorophore. And every time the complementary nucleotide actually binds, it fluoresces, and then essentially the next-gen sequencer just captures an image. Like it's just capturing one image, and then another nucleotide comes, it captures another image, and another nucleotide comes, and it captures another image. So you just have all these series of fluorescent images, and essentially there's going to be a specific XY coordinate where this particular cluster is, right? And so the instrument, the sequencer, is just tracking that specific coordinate, XY coordinate, and it's just tracking, oh, what every image, like what's the nucleotide being added? And of course, in reality, that's happening simultaneously, like billions and billions of clusters on a given flow cell that the sequencer is imaging, and you capture all the sequences like that. And I mean, that's essentially next-gen sequencing in a nutshell. And from there, we generate all of these sequences. Like I said, they'll probably be 200 to 300 bases in length. And this will be in the form of a FASTQ file. So that's in general what we deliver to you, a FASTQ file, and you give that to your bioinformatician, and this is what they're doing with it. Like they're aligning it probably to itself, but if you have a reference, then hopefully align it to a reference, right? I mean, obviously, if you don't have a reference, you have to do de novo assembly, and that sounds like a nightmare, but anyway, there's that. And likewise, like, maybe you'll look at the reference and say, hey, like, how come my sequence is a little bit different from the reference? How come on the reference it's an A, but on my sequence it's a G? I mean, well, congratulations, you found a mutation. So that's essentially how that works, right? Okay, so now that we really went over like 3 key technologies, methylation array and bulk RNA-seq and next-gen sequencing, this paper here by John DeSisto and Adam Green, they've applied these techniques, and we'll see how they applied the techniques and see what they discovered. And the title is Comprehensive Molecular Characterization of Pediatric Radiation-Induced High-Grade Glioma. It's a mouthful, and every one of these papers are going to be a mouthful, so just get ready. Yeah, Nature Communications, so it's pretty high impact too, so good for them. So let's kind of go over what this is all about. So a little bit of background. So therapeutic radiation of the central nervous system to treat childhood cancers can cause secondary damage to form new radiation-induced tumors known as radiation-induced glioblastomas. So they're called RIGs. And RIGs arise in 3% of pediatric cancer survivors after cranial radiotherapy, and is a significant cause of late mortality in childhood cancer survivors. And sadly, there are limited therapeutic options. And so the approach that DeSisto et al. Took was to collect tumor tissue specimen from fresh frozen fresh sections, FFP tissue. What in the world? Oh, okay. Yeah. Frozen sections, FFP sections, and frozen patient blood samples. And then they proceeded to conduct comprehensive molecular profiling of these RIGs, okay? And like I said, the assays that were specifically performed by the GSR with methylation array, bulk RNA-seq, and next-gen sequencing. And essentially, here are some of the results that they got. And wow, that looks really busy, doesn't it? So let's take a closer look at some of these figures one by one and see what's going on there. So taking a look at the first one, what you're essentially seeing here is a circular dendrogram, and that just basically shows the hierarchical relationship between the objects. And essentially, they use the dendrogram in order to generate a reference based on the CNS tumors, and the data that they're using are the methylation data, right? So they managed to take these kind of well-characterized methylation groups associated with CNS tumors to generate this reference, okay, color-coded as well. And essentially, when they overlaid their methylation array results, they show that relative to the reference CNS tumors, 25 out of 31 RIG cases clustered among a group of epigenetically similar tumors known as PET-RTK1 and GBM-mid, right? So there it is, like here's the reference, right? And then if you look closely, you see the red and the light blue right there. So those correspond to the PET-RTK1 and the GBM-mid, and 25 out of 31 RIGs are all clustering together. And this is based on their methylation array data, and so this is a very interesting finding, because these RIGs are actually coming from all of these disparate clinical origins. And yet, when you look at the methylation data, it's showing that these RIGs are predominantly aligning with a specific methylation subgroup, the PET-RTK1 and GBM-mid. So that's interesting. Like how could it be that all these RIGs are coming from all these different patients, but they have a very similar methylation profile? So that's kind of their first clue that there's something interesting going on here. There's some kind of commonality between these RIGs. This is a secondary method in which they analyze the data. This is with the t-SNE. I think many of you are already familiar with it. It's just a dimensionality reducing way of kind of visualizing the data. But long story short, the closer these clusters are together, the more similar they are, and the more further they're apart, the dissimilar they are. And in this case, they're looking at the methylation group. So here's just a secondary analysis of their methylation data. And likewise, you got the PET-RTK1 and the GBM-mid kind of clustering right there. And then the 25 out of the 31 RIGs are clustering together. So here's a secondary way that they've analyzed their data, just kind of once again recapitulating the finding from the previous with the dendrogram, in order to show that these RIGs, there's something going on there. This is also the dendrogram, but this time they're actually showing a full heat map of their findings right here. So obviously we have no idea what's going on here, but out of the 850,000 different different biologically relevant targets. They're showing 20,000 of them. And likewise, same, the same idea, but now you get to actually visualize the methylation, the results basically over here. So once again, 25 out of the 31 rigs are clustering here. And this is essentially where the PET RTK1 and the GBM mid are in the reference. From there, they've also, so that's essentially their methylation data. They also looked at the transcriptomic data as well. So I have to confess, it's not just RNA-seq that they got the transcriptome data from. They also used microarray. So, that's kind of an outdated technology. Once upon a time, there was no bulk RNA-seq, so you had to do microarrays in order to get gene expression. But anyway, that's what they had, so it is what it is. So what they found was that, well, first of all, the transcriptomic data were only available for 13 rigs cases, but okay, that's fine, and 42 de novo glioblastoma cases, right? So let's once again take a closer look at what's going on there. What they found was that these rigs are clustering separately from the GBMs. So what does that tell us? Well, that tells us that these, looking just from not the methylation data, but rather coming from the transcriptomic point of view, these rigs also have a unique characteristic that seems to distinguish them from all of these other different GBMs, right? So that's a secondary clue that there's something common about these rigs essentially happening here. But what's kind of cool here is that if you look at the actual t-SNE itself, you'll notice that there's actually two subclusters. So they're actually, they're obviously in the lower right hand, so they're similar to each other, but there's actually subtle differences between them. So they're clustering a little bit separately. And it turns out these rigs, they form two subgroups, A and B, and they're driven by 2,961 genes. So that's what you're seeing on the bottom there. There's 2,961 genes. There's a heat map, and it looks like a little bit more than half of those genes are downregulated for group B, but in contrast, upregulated for group A, and vice versa, upregulated here, but downregulated right there. So yeah, I mean, so that's that, but so when we kind of look at that methylation clusters again, right? So earlier we've already determined that these rigs are very similar to the PET RTK1 and GBM mid subgroups. So sure enough, the methylation clusters alone cannot distinguish rig from PET RTK1 and GBM mid, right? So no surprise, that's what I talked about in the previous slides. But then when you actually overlay, right, the transcriptomic data onto the methylation clusters, now you can in fact distinguish the RIG group A and B. So in combination, it turns out the DNA methylation data is matching the transcriptomic data for the subgroups A and B. So here you'll notice that 5 out of the 7 group B, which is in red, they're kind of on the left side, and the blue 5 out of 6 of the group A are on the right side. So now the transcriptomic data is actually revealing that there's some kind of subtle differences between these two groups, subgroups of these rigs. And when they took a closer look at those 2,961 genes, they essentially analyzed the data via gene set enrichment analysis, GSEA, and that will allow you to interpret gene expression data by focusing on the gene sets, so the group of genes that share the common biological function, right? And what they found is that group A rigs have stem-like proneural and neuronal characteristics and enriched for MYC pathway genes. Okay, so there you go. And group B rigs have mesenchymal and astroglial characteristics, and importantly, relative to group A, they have decreased DNA repair gene expression. And that ends up being really important. So here you get the DNA repair, the group A relative to group B, it's just more enriched. So, and then what they proceeded to do was take a look at the germline as well. So briefly, the FDR, I guess that's some kind of statistical test, and the closer it is to 0, the more accurate or statistically significant your results are. And essentially what they found is that you can recapitulate this data from not just the somatic, but also the germline as well. And likewise, all these normalized enrichment scores seem to essentially indicate that this group A is relative to group B is enriched for DNA repair, but like I said, in contrast, group B it's downregulated for DNA repair. Okay, and so taken into totality, there's a handful of conclusions that DeSisto et al. Can essentially derive. Number one, well, DeSisto et al. Evaluated the largest RIG cohort to date through comprehensive clinical and molecular analyses. The DNA methylation profiles reveal RIG tumors to having defining characteristics independent of their clinical origin. The transcriptional profiles distinguish RIG from de novo GBMs, right? They all cluster together. What that tells us is that some treatments may be effective for RIGs, but not effective for de novo GBMs, and specifically pediatric glioma, because that's what this is all about, and vice versa. So that's something to kind of take into consideration for therapeutic purposes. You have to know that. And here's another kind of interesting point, which I think is highly impactful. Insights into the germline susceptibilities in group B patients related to impaired DNA repair might help identify patients at risk for RIG before tumor therapy. So in my mind, like that tells us that there's kind of immediate clinical ramifications to these findings where, maybe you can do, follow their lead and, take some of these cells and you do to maybe some kind of sequencing, whole genome, whole exome, or maybe enriching for these DNA repair genes. And if you are among these group B patients that have impaired DNA repair, you can just say, oh, you belong to this group that's susceptible for RIG, so we're going to do a different tumor therapy than what we would do traditionally. So I think that's great that they have this finding in this paper. So all the credits to John DeSisto, Adam Green. If you have more questions about this, by all means, please reach out to them. So let's move on to the next technology and then the next paper that we'll be talking about. We're going to talk now about single-cell RNA-seq, okay? So what is single-cell RNA-seq? Well, single-cell RNA-seq examines the sequence information from individual cells using next-gen sequencing. So starting from a heterogeneous cell population, you can investigate cellular differences in the context, differences to better understand the function of an individual cell in the context of its microenvironment. And so what you're seeing at the bottom here is sort of a representative workflow of what single-cell RNA-seq would look like, starting with single-cell capture, followed by high-throughput sequencing, next-gen sequencing, gene expression quantification in the form of a heat map here, followed by population visualization in the form of UMAP or T-SNEs, and then depending on if you can kind of see the trajectory, like pseudo-time trajectory, like maybe there's some kind of hierarchy in your single-cell data, you can actually infer the trajectory. Maybe these are differentiating in some way, and you can get that information. And I bring that up because not so much for single cell, but for the third paper I'll talk about, they actually kind of use this too, the idea of inferring some trajectory. So I'll just kind of keep that in the back of your mind. So how exactly is single-cell RNA-seq performed? Well, virtually all single-cell RNA-seq involves barcoding individual cells as the first step. And so specifically all transcripts in any given cell is labeled with the same barcode. And on the right, what you see here is sort of a representative figure of what that process would look like, where let's take this. So this is a oligo, and this oligo here has two important features. Number one, you got the poly-DT portion right there, and then you have the cell-specific barcode, and we'll just call this the green barcode here, right? And essentially, this oligo is going to act as a primer for reverse transcription. So as that poly-DT portion hybridizes to the poly-A tail of the mRNA, it'll undergo cDNA synthesis, as it just follows along, making a complementary strand to the mRNA. But that cDNA is going to be conjugated to this green barcode. And the idea is that every single cDNA synthesized inside this cell is going to have a green barcode associated with it. Whoops. And every single cDNA synthesized inside this cell is going to have a magenta barcode associated with it. And every single cDNA inside this cell will have a purple barcode associated with it, et cetera, et cetera. And during the NGS next-gen sequencing, all these transcripts from all these different cells exhibiting all these different barcodes are pulled and sequenced simultaneously. But during the bioinformatics stage, the individual cell information is demultiplexed from the pool using the barcode label. So your bioinformatician will, let's say, look at gene A and say, oh, I see gene A has the green barcode associated with it. It must mean it came from cell number 1. And I see gene B that has a purple barcode associated with it. It must mean it came from cell number 3, et cetera, et cetera. So that's essentially how you get the single cell information from. Of course, that sounds all fine and dandy, but how in the world do you actually ensure that each of these oligos with the specific barcode is within a specific cell? So believe it or not, there's actually many ways to do that, but the specific technology that we use is through a process called DropSeq. So DropSeq, the main purveyors of this technology, is a company by 10x Genomics. And here, the magic formula is actually these gel beads. So these gel beads, they have essentially oligos sticking out of them, just like you have hair sticking out of your head, or at least for some of us. And essentially, if you look at the oligo, you got the poly-DT, there you go, as I mentioned before. And then you've got that here, the cell-specific barcode, or they just call it a 10x barcode. So we'll call this a green barcoded gel bead, right? And so this gel bead will have a yellow barcode, this one will have a blue barcode, et cetera, et cetera. And the idea is to put this into a microfluidics instrument where we're flowing simultaneously the gel beads and the cells into the microfluidics instrument in order to create these micro droplets surrounded in oil emulsion. So ultimately, at the end of all this, what we're going to have is a tube of oil, and inside the oil, you have all these tiny little droplets, okay? And the goal of this is to ensure that inside a droplet, you get one cell and one gel bead. And each of these droplets are going to act as a micro reaction, basically just all happening simultaneously, where inside this particular micro reaction, let's say, remember, this gel bead has red barcode. So every single cDNA synthesized inside this cell is going to have a red barcode associated with it. And every single cDNA inside this cell is going to have a black barcode associated with it. And every single cDNA inside this cell is going to have a purple barcode associated with it. And of course, there's the instrument that actually does all this. Here's kind of a cool little GIF that I found. And if we follow along this cell in the red arrow, here it comes, the gel bead's coming from the left, the cell coming from the bottom. And if we follow the mouse, you'll notice here's a droplet with a cell and a gel bead right there. So that's essentially how microfluidics, the drop-seq works, and that's what it's all about. And here's actually a picture of a PCR tube that I took with my phone, which also speaks to the amazing resolution of our phones. And I don't know how easy it is to see it, but if you, I don't know, on my computer, you can see it. I don't know if you can see it here, but you can actually see the little tiny little micro droplets. I mean, I don't know, like they're there. Yeah. So that's really cool stuff that you can see these actual little droplets in the tube itself. So good stuff. So here we go. Now, Kent Ramandi and Rajiv Vibhakar, they used a single-cell RNA-seq for their research, published in Neuro-Oncology, so another great high-profile paper. Here is their title: Neoplastic and Immune Single-Cell Transcriptomics Define Subgroup-Specific Intratumoral Heterogeneity of Childhood Medulloblastoma. Yeah, okay. So yeah, let's see what's going on here with their findings. So a little bit of background. Medulloblastoma, MB, is an aggressive childhood brain tumor comprised of 4 consensus molecular subgroups: GP3, GP4, SHH, and WNT. And Ramandi et al. Aim to determine the influence of neoplastic and immune cell diversity on medulloblastoma biology in 28 patient samples. And so what they did, they collected 28 human MB samples from surgeries. And what makes this paper unique is that compared to all the previous studies, they use a greater number of samples and cells per tumor to perform single-cell RNA-seq. And of course, like how we contributed is by performing the single-cell RNA-seq and next-gen sequencing for them. So there's that. And here we go. Like, let's take a look at some of these results and see what their findings are all about. Another really, really busy figure. So we'll take a look at these a little more closely, right? So here, what you're seeing, first, let's kind of go with the obvious ones. Here are the 28 patient tumors. They're individually color-coded with the numbers. So when you see all these colors, each color essentially denote a specific patient, okay? And what's kind of cool is that when they took the single-cell data, they generated the UMAPs, indeed, in line with the kind of the well-established subgroups for medulloblastoma, they seem to segregate accordingly, like at the GP3 population, GP4, SHH, and WNT. But honestly, I'm getting ahead of myself because even before that, they actually managed to cluster them based on the neoplastic, right? The tumor-like neoplastic clusters all the way over here. The non-neoplastic, they're all the way down here, right? And then among the non-neoplastic, you got the substantial immune population down there. So essentially, they're all clustering together. And like I said, within the neoplastic, you got the GP3, GP4, SHH, and WNT. So far, so good. I mean, just from like, let's say a QC standpoint, this is kind of in line with what's in the existing literature. So at least getting this far to recapitulate the data tells us that their data is probably good, right? So that's essentially the UMAP, but well, here's where things get a little bit interesting. They actually use an algorithm or a program or I don't know. But anyway, it's called InferCNV. And what InferCNV does is it looks at differences in gene expression compared to reference normal cells to infer gain or deletion of chromosomal segments, i.e. copy number variation. And you know, if you think about it, it's actually quite simple but brilliant because you can imagine like just from the single cell data, right? Single cell RNA-seq data, if you see like a group of genes being downregulated and those genes just happen to come from like the same region of a chromosome, then you can infer that that region of the chromosome is deleted. But likewise, if you see like a group of genes that are overexpressed and they all just happen to come from the same region of a chromosome, that you can infer that maybe that region of the chromosome is duplicated. So that's essentially the InferCNV in a nutshell. And what they found is that even not, so this isn't a UMAP anymore, this is InferCNV. Apparently the data is also consistent in terms of clustering these samples together along with those subpopulations that I told you about earlier. You got the GP3, GP4, SHH, and WNT. So once again, kind of using a different bioinformatics method, but recapitulating that difference also gives us confidence in their data. But they also find some other novel, interesting things too, where, for example, at least among the GP3 and GP4 population, this isochromosome 17q is exhibiting MB subgroup specific CNVs and GP3 and GP4. So there it is. Like it looks like for chromosome 17 on the front end of things, there's a lot of deletion going on, and you know, on the tail head middle to the tail end things, there's a lot of duplication going on, and that seems to be kind of characteristic of GP3 and GP4 subpopulations. And I mean, that's great. Like they can kind of figure that out using InferCNV, not just from a UMAP, but from this. So that's really interesting stuff. And what they do is they proceed to actually find another kind of interesting correlation with this data. So for example, let's kind of start over here. These are CNV counts, meaning how many actual CNVs are there in any given cell. And some obvious ones, let's say the bottom is 0 in gray. And for non-neoplastic cells, there's no CNVs. I mean, big surprise, right? They're not neoplastic. So, okay, that's great. Like they're just like kind of normal cells for the most part. So there they are, a low CNV count. But then the CNV counts, the colors here from light to dark indicates how many CNVs there are. And let's kind of look at this for just real quick here. So let's take GP3, for example. There's 2 patients in this specific cluster, right? You get the blue and the pink, so that's 2 patients right there. 1, 2, 3, there's 3 patients right there. And essentially that's what all these numbers mean. 2, 3, there's 6 patients in this one, 4 patients in this one, 7 patients in that one. What they find is that apparently there's an interesting correlation between the sum of the CNVs, where the lower the CNV counts, they're colocalized with the patients. So like, for example, over here, like these are basically like the same, right? Like 7, 7. This group of clusters over here, there's 7 patients in there, and all 7 patients just happen to have low CNV counts, and 6 patients low CNV counts. But then the handful where there are high CNV counts, so like 12 to 16, they're all clustering over here, and they're kind of also like patient specific. Like there's only 1 in each of these groups. So that's kind of another interesting correlation that they found using infer CNV to determine like something along those lines between the samples and the CNV counts. So that's interesting stuff there. Now, what they proceed to do is now look at the immune cells, actually. So among that non-neoplastic population, remember there's some immune cells, right? Raimondi et al., they took a closer look at that. And the 2 main clusters that they found among the immune cells were the myeloids and the lymphocytes. So the lymphocytes, kind of obvious. You got the NKs, the Ts, the Bs, and the Tregs, right? But what was interesting is that unexpectedly, among the myeloid population, it was surprisingly diverse, where you have all of these kinds, like non-activated microglia, complement myeloids, M2 activated myeloids, chemokine myeloids, dendritic cell-like myeloids, and neutrophils. So what's going on there? Well, so this is, there's one in particular that turns out to be very, very interesting. So they look at all of these different subpopulation of the myeloids, but this one in particular, the M2 activated myeloid, was unique. And what made them unique was that it seemed to be the case that these M2M was discovered to be more abundant in that SHH subgroup than any other subgroup. And basically, the y-axis, that's actually the number of patients. So in that specific SHH cluster, there were 9 patients, and 6 of them, you can kind of tell like they're above the error bar here, just really, really statistically significant, and 3 down here. But even then, you might argue that out of the 3, like 2 out of the 3 are kind of on the upper end of, the M2M, like activated, let's say samples and whatnot. But anyway, but then here's kind of where the beauty of single cell RNA-seq comes in. So there's actually, within these subgroups, there's actually subtypes, right? So for example, among the SHH subgroups, there's actually 4 subtypes: alpha, beta, delta, and gamma. And essentially, these are all nomenclatures that are well established in the field. But beta and gamma correspond to the infant SHH subtype, whereas the alpha and the delta correspond to the adult or adolescent SHH subtype. And thanks to single cell RNA-seq, what they find, so what you're seeing here, all of these dots just kind of represent a single cell. And from there, they've identified that this M2M enrichment is not just ubiquitous across all SHH samples, but rather in this subtype of SHH subgroup among the SHH beta and the SHH gamma. And like I said, given that these 2 subtypes are originating from the infants, what they kind of conclude, or at least kind of infer from this data, is that M2M may be recapitulating age-related neurodevelopmental functions rather than an immune role, given their abundance in the infant SHH subtype. So again, like you wouldn't be able to tell this with bulk RNA-seq, and in fact, like you might actually get into trouble just by using bulk RNA-seq, where you find out, hey, look at that, like M2M is upregulated among SHH subgroups. I'm going to publish it, but then when you actually use single cell RNA-seq data, you find that, okay, well, it's not just across all SHH subgroups, but rather specifically in the beta and gamma. And that's going to be really important because, M2 activation, like, associated with the immunosuppression, et cetera. So that kind of gets to the last point, but essentially, okay, well, I'll get there. So Raimondi et al., so here are some of the conclusions. Raimondi et al. Discovered that sample-specific clustering in MB medulloblastoma single cell RNA-seq is influenced by CNVs. right? So we went over that. They identified considerable immune cell diversity in MBs, including subgroup-specific immune populations, such as SHH-associated M2 myeloid cell population. And taken into totality, what they propose is that the therapeutic strategies targeting the specific immunophenotypes of MB are necessary and can be advanced based on the findings of this present study. And where I was going with that earlier was that, if you see M2 activation, maybe they're more resistant to immunotherapies. So you just have to take that into account before you even, like do your traditional immunotherapy and get confused, like how come it's not working? I mean, maybe they belong to the beta and the gamma subtype. So it could be that. So anyway, I think these findings are great. There's Kent Ramandi and Rajiv Vipakar, all the credit to them. So yeah, we'll see. We'll see where all of this goes going forward. The last technology and the last paper I'm going to talk about is spatial transcriptomics. So let's kind of go over what exactly is spatial transcriptomics. Then there's many different kinds of spatial transcriptomics, but the one that I'm going to specifically talk about is called Visium. And Visium, what the magic formula here are these slides. So this is a Visium slide, and on the Visium slide, you have these squares, what are capture areas. And if you zoom into a capture area, you have these spots. Okay, and if you zoom into a spot, what you're going to find are these oligos. And on these oligos, you're going to have the poly-DT, and this time, not a cell-specific barcode, but a spatial barcode, right? So what it tells us is that this green spot over here is going to have a green barcode associated with it, and this blue spot over here has a blue barcode, and this red spot over here has a red barcode, et cetera, et cetera. And so that's the Visium slide, right? And then next, we have the tissue block. We section the tissue block in order to cut a little section right there, and then directly transfer that section onto the Visium slide, okay? And then we take that Visium slide, we actually do H&E staining and imaging, followed by permeabilization so that the tissue actually becomes permeabilized, it becomes porous. And then through the pores, you got the mRNA that is going to seep through the pores and gets kind of captured by these spots. And essentially, these spots, remember those oligos, they're going to act as primers for reverse transcription. So every single cDNA synthesized on this spot is going to have a green barcode associated with it. And every single cDNA synthesized on this spot will have a blue barcode associated with it, et cetera, et cetera. And following that, of course, we make the libraries, we sequence, but then the bioinformatician will look at that and say, oh, I see gene A has a green barcode associated with it. It must mean it came from this section over here on the tissue, like all the way over here. And I see this gene B that has a red barcode associated with it. That must mean it came from all the way over here on the tissue. So that's how you preserve the spatial information. Another way I kind of like to think about this is where, on the capture area itself, there's actually 5,000 spots. So let's say you actually had a tissue that covered the whole capture area, right? You're essentially doing 5,000 bulk RNA-seq simultaneously from the tissue, and then overlaying those gene expression on where on the tissue they're coming from, because all those barcodes are already predetermined. They know precisely which spot has which barcode and where it's located. So that's essentially spatial transcriptomics in a nutshell. And in this next paper, also on uro-oncology, Ray Fu and Andrew Donson applied spatial transcriptomics to their research. This one, another mouthful. Spatial transcriptomics analysis delineates epithelial and mesenchymal subpopulations and transition stages in childhood ependymoma. So what in the world? So let's take a look. So some background. Ependymoma posterior fossa subgroup A, PFA, is the commonest and deadliest childhood brain tumor EPN variant, and sadly, it exhibits therapeutic resistance. Previous single-cell RNA-seq results revealed a cellular heterogeneity of EPN tumor microenvironments, and what Fu et al. Aim to do is determine the spatial distribution of the different cell types within the tumor microenvironment. And the approach they took, well, they collected 14 surgical tumor sections. Then they utilized spatial transcriptomics to map the transcriptomic data onto the tumor cellular architecture in PFA. And how we at the Genomics Shared Resource helped was by performing the spatial transcriptomics experiments and next-gen sequencing. So once again, some more busy figures here. So let's take a closer look at what's going on. The first things first. So what they did, they had 14 samples, and then essentially out of the 5,000 spots on the capture area, about 2,400 spots were actually under the tissue. So it looks like their tissue was kind of half the size of the squares. So they got 2,400 spots per sample, 14 samples, and that just kind of totaled to 33,082 spots. And then when you generate a UMAP from all of these spots, from all these 14 different samples, they've identified 23 clusters, of which 17 clusters were present in all 14 samples. And, again, kind of recapitulating some of the things we found earlier, you got the non-neoplastic population, and then you got the neoplastic populations up there. And it turns out among the neoplastic population, you got two specific clusters. You got the mesenchymal and the epithelial, right? And so that's also good. That's consistent with what we would expect from the data. But then this is from a UMAP. They also took the data and underwent this algorithm called harmony alignment. And what this does, harmony alignment, it's an algorithm that groups cells based on cell type rather than data set specific conditions. So now, maybe you have all these different samples coming from all these different patients, and there could be a lot of differences between these patients and a lot of noise consequently. But what this algorithm will do, it'll prioritize cell types during the data analysis. So that's essentially what they used. And what they found is that there's two major branches among that neoplastic population that comprised of either epithelial or mesenchymal clusters. So great. I mean, like I said, two different methods to analyze the data, and they're coming up with similar conclusions. So once again, that's good. That's in line with what we would expect, hopefully, and the data is kind of consistent. So that's great. But oh yeah, there it is. So there's the epithelial and the mesenchymal. But here's where the beauty of spatial transcriptomics comes in. What they found is that when you then take those epithelial and mesenchymal clusters and actually map it back onto the tissue itself, so broad visualization, what they find is that the epithelial or mesenchymal clusters on the tissue landscape is spatially distinct or segregated into zones. I mean, this is my point. Like, without spatial transcriptomics, you wouldn't know this. Like, you might think, oh, okay, all those green and red spots are all over the place on the tissue like a mosaic pattern. But thanks to preserving that spatial information, now we know that these zones are distinct and segregated. And honestly, the paper actually goes into characterizing both zones, but in the interest of time, I'm only going to talk about their findings from the epithelial zone. So there it is. But yeah, so again, with single cell, you can't do this. I mean, look, I love single cell, but like spatial, just preserving that spatial information that really reveals a lot of novel things here. So they do some really interesting stuff based on the spatial data. Remember, it's not just a transcriptome, but there's also an H&E image that comes along with spatial transcriptomics. And essentially, when we start taking a closer look at this, so there's the epithelial zones. It turns out they were comprised of 6 different clusters. You got the UECA, so that's the undifferentiated ependymoma cells, and TEC A, B, C, D, so those are transportive ependymoma cells, CEC, so ciliated, and then they get the M in the mesenchymal zones. Obviously, they're not going to be here, but you get the MECs, right? And here's kind of the first interesting bit of clue that they found that was interesting, that the UECA spots are associated with denser cellular areas. So if you kind of look here on the H&E stain, where all those purple spots just kind of represent nuclei, they also used purple for whatever reason. The UECA just seems to be colocalized with those dense cellular areas, right? Where there's a lot of nuclei. That's also where the UECA subgroup of the population, the clusters are, these cell types. So that's interesting. So that's like kind of the first clue that maybe there's something interesting going on here with the UECA. Then they generated UMAPs from these cells. And likewise, what they found was that there was a cellular hierarchy essentially associated with this. And using some pseudotime trajectory analysis, they found that hierarchy initiating from that UECA, so the undifferentiated ependymoma cells, all the way up there by itself. And then you can kind of map out what is going on, how are they differentiating or something. And it looks like adjacent to it are the TECs, the transportive ependymoma cells ABC. And then that proceeds to branch off into these other zone clusters. So you got the CEC, you got the TEC-D, and then you got the mesenchymal zone cells. So once again, a secondary analysis using UMAPs are essentially telling us that the UECA, there's something special going on there, something very interesting. So that's clue number 2. From there, they actually looked at the gene expression profile a little bit more. And again, it turns out a single cluster, UECA, was characteristic of undifferentiated EPN cells. So these genes, ELN, MFAP2, MDK, and COL9A2, I guess they're characteristic of undifferentiated cells, so that's what they found. They also exhibited high protein synthesis, that is to say enrichment of ribosomal gene expression, and most importantly, proliferation, cell cycle. So if we take a close look over here, you'll notice, I mean, by the way, like, high school biology, it's been a while, right? G0 is associated with cell cycle arrest, G2 and M, mitosis, like the cell division, and the S phase, the DNA replication, the synthesis, right? And here you go. The UECA, this cell type over here, was characteristic of cell division. So in this heat map, upregulated for cell division, down-regulated for cell cycle arrest, and kind of neutral in the DNA replication. So there you go. Now there's a third piece of clue that's just telling you that there's something really interesting going on with this UECA cell types. And essentially, the paper proceeds to kind of characterize it further, but one way or another, here's kind of the conclusions that they derive. So Fu et al. Essentially discovered that the UECA is a highly proliferative epithelial zone progenitor cell that is associated with progression in PFA. So I thought this was kind of the best place to put this other figure from their paper here. They actually followed up with the results, looking at clinically annotated bulk gene expression data set. So looking at the outcome of these patients, progressive or non-progressive after 5 years. And what they found was that this data of the characterization of UECA was consistent in associating UECA enrichment with PFA tumor progression. So here we go, like on the top row, that those are progressive, tumor progressive patients, and here's a non-progressive. And among the progressive patients, you'll notice that the UECA is specifically enriched. So, you take all of these kind of things together, what they essentially discovered that this UECA is really important, and they proposed that UECA may be a novel target for therapeutic development in PFA. So all the credit goes to Ray Fu and Andrew Donson. I mean, so if ever it comes out that, they found some kind of drug or treatment that targeted UECA, like, where that idea came from, like this paper right here using spatial transcriptomics and all these different technologies. So really good stuff. So here's some of the takeaways that I took from going over this. Like I said, it was really gratifying. Usually I'm on the technology side, and sadly I don't really get to see what the outcome is. And but I thought it was really incredible. But here are some of the points. Basically, hopefully I've done a good job kind of convincing all of you that all these various advanced genomic technologies can, in fact, be applied towards cancer research. And really, there's like dozens of papers I could choose from, but I thought these were good, and a lot of pretty figures in there too. So wanted to showcase them. Advanced genomics technologies generate a lot of data that requires a combination of deep expertise in the subject and creative bioinformatics capabilities. And again, this is kind of my blind spot, right? Like, obviously I can help you get your data, like high quality data, but that's up to you to have deep expertise in the subject itself. And, obviously if you're a bioinformatician, great, but if not, making sure that you have somebody who can analyze the data, looking at this huge data set from all these different angles so that you can kind of make some novel discoveries. I mean, who would think to use in first CNV, like, that's really cool stuff. So I think having a really good bioinformatician is going to be really, really important. Numerous CU Anschutz investigators are taking advantage of the GSR technologies to expand our collective understanding of cancer. And again, hopefully I've done a good job of kind of showcasing that for you as well. And these cancer-related findings often have therapeutic and/or clinical significance. And I always, like I said, for all of these papers, that's kind of a tagline that I want to kind of include. Like, what are some, like, these aren't findings that are just going to end up in your textbook. Like, these have real potential for therapeutic and clinical significance, and how can it be translated in that sense? And yeah, I think that's the case. So with that said, none of this would be possible without the wonderful team that we have at the Genomics Shared Resource. Like, they're the real MVP of all of this. I mean, I'm just here to talk about it. Like, they're the ones really making the magic happen. So they really deserve all the credit. So thank you very much to them. And honestly, thank you so much for having me today to present for you. It's been an honor. Thank you very much. Yeah. Yep.
I think adenosine also can be methylated. So adenosine.
I guess I'll start over.
Yeah.
So with the methylation array, you mentioned 5-methylcytosine being which you can use to look at all the different markers.
That's a good point, and you're right. I mean, a lot of, by the way, between you and me, like, not just for the methylation array, but even for bulk and single cell and spatial, these were just sort of representative kind of simple diagrams to illustrate the points so that everybody just understands the technology behind it. I mean, so for example, even with spatial transcriptomics, we don't actually generate cDNA via reverse transcription anymore. We use probes, and I just, I kind of lowered the level down. Obviously, a lot more about methylation rate than the average person, but I just really wanted to make sure everybody just kind of understand the underlying simple point about how these technologies work. But you're absolutely right. I think there are far more complexities with the technologies.
Oh, okay. So yeah, I was hoping if you could. I don't know about methylation arrays, so I was wondering if you could let me know if adenosine methylation affects what you may see for because you talked about cytosine.
Yep.
I think.
So my understanding is not really. Like, somebody should fact check me on this. But my understanding is that you have these CPG islands, and those are kind of the hotspots for methylation with the cytosines. So I think that's what the arrays specifically investigate. As far as those 850,000 biologically relevant targets, like that's where the sequences are originate from. Like I said, I'll need to look in a little bit deeper. This is also kind of my blind spot that you just uniquely kind of zoned into to point out. But yeah, I mean, that's a great question. I think it's worth following up on. But to my understanding, it's really the cytosine that is most impactful, especially in a mammalian context. And I'm 99% sure that's specifically what the methylation arrays look for. Yeah.
Thank you for a nice overview of all those methods.
Yep, thank you.
My question deals with the spatial transcriptomics.
Mm-hmm.
Could you please compare available technologies? So you talk about the Visio. I believe that there will be, if not already present on the campus, also other instruments like NanoString on the campus. Could you please compare the available technologies, maybe not only those available on the campus, in terms of how many cells can be detected, how many RNAs on average, and so on? What are the options and what would be your recommendations? Thank you.
Oh, yeah, absolutely. And, you know, honestly, that is, you know, usually the talks I give, that's what it's usually about, you know, the related technologies. And, you know, like you should come to some of my talks next time too, you know, because we have even more new spatial transcriptomics technologies coming out. At the moment, basically, we have two different spatial transcriptomic technologies available. Number one is through 10x Genomics, that's the Visium that I've kind of showcased here. The other is, yeah, as you correctly pointed out, NanoString. And NanoString actually two different kind of technologies. The Genomics is the one that we've really have experience with for almost 2 years now. And we like to say that Visium is a discovery approach because it just gets a transcriptome profile across the entirety of the tissue, versus Genomics is a targeted approach where you're actually zooming into a specific region and then kind of getting the transcriptome profile of that region. So those are the two kind of well-established ones that are core. We're getting some new ones where, which, you know, okay, so without getting into too much detail, there's basically two different branches of spatial transcriptomics, but basically the trade-off is that one can do the whole transcriptome, so all like around 20,000 genes, but they're not single cell resolution. But then you have another branch of spatial where it's single cell resolution, but you can't investigate all the transcriptomes. So maybe at most you're looking at 1,000, maybe 6,000 in the very near future. And essentially, like the holy grail of spatial transcriptomics for now is doing single cell resolution and full transcriptome. And in that sense, there's Visium is working on that. They're going to hopefully launch Visium HD, so that'll be single cell resolution. Cosmics, which is the other NanoString technology, we just finished our training last week, so that's going to be that's in the works. Basically, we're going to internally validate it to make sure the technology actually works in our hands. That one is not just single cell, but subcellular resolution, but it's not the full transcriptome. At the current iteration, it can look at 1,000 RNAs, predominantly used for cell type identification. But then like the next version is going to come out to 6,000 plex. So now looking at 6,000 RNAs, and apparently those RNA targets are specific for like a certain pathway. So obviously 6,000 out of like 18,000, that's not the full transcriptome, but those are kind of pathway specific RNAs. So they can, not only can you do a cell type identification, but you can infer the pathway as well. And I would say like, it really kind of goes down to those three technologies. There's more that's kind of a variation of the Cosmics. So, you know, 10x Genomics has one called Xenium. They do something similar, but they don't look at thousands. They look at dozens to hundreds. And, you know, but again, it's like probe-based, also single cell subcellular resolution, but not the full transcriptome. But yeah, basically, there's a lot there. And if you want, you can reach out to me and I'll be happy to talk to you one-on-one more about these spatial transcriptome technologies. But that's kind of the simple overview. Yep. Oh, yeah.
So for the spatial transcriptomic, how many slides do you normally examine per tissue?
Oh, so basically,
My question is this bidimensional or can you do three-dimensional? Because if you do only one slide, that's not really representative, right? Because you may be in this area of the tumor, but not here, not here. So how many slides normally would you do for this analysis?
No, absolutely. So that's a fantastic point. That's actually been a major kind of drawback to spatial transcriptomic technologies up to this point, where, you know, we would take a tissue block and we're sectioning it, but how deep into the section do we actually know that we want to investigate, because you're right. It's, you know, like, how would you know? So I didn't talk about it here because, like I said, the focus of this talk was more on the research side rather than the technology side. For the past few months, we have this new instrument called the Site Assist, which actually allows you to transfer, again, kind of speaking loosely, transfer a section on a generic glass slide, a standard glass slide. So how that translates to is basically, let's say you have a tissue block, right? And let's say you just keep cutting sections. You put one section to cut the section, put it on the glass slide, put another section, put another glass slide, put another section, so let's say you do 20 sections on 20 separate glass slides, right? From there, you can do, let's say, H&E staining or some kind of immunofluorescence staining to figure out specifically which section that you're interested in, and only use that one for the Visium assay. So,
that would be really biased. I think to be fair for this technology, you should do like most people do for pathologies. You cut one, then you discard maybe 3 or 4, get another one, and get the overall that you can actually construct the tumor itself, if it's a tumor or any other tissue. Because if you go and you select, then that's not going to tell you information. But that's what I say, I don't know how difficult it would be to have like maybe 6 slides for tumor where you do one, then cut it, discard those, get another one. That's what it normally is called series sections. So
I mean, I don't disagree at all. I mean, I think the only bottleneck at the moment is the cost. Like, there's honestly nothing stopping.
that was my second question.
I see. No, yeah. Each of these experiments are like, just, you know, the slide themselves are worth like $3,000 for 2 sections. So, you know, if you do like multiple sections on a given block, like, you can already imagine. But I guess, you know, now that you put it that way, the real holy grail is single cell resolution, full transcriptome, and not just like a 2-dimensional section, right? It's really the whole tissue block. No, I mean, and by the way, like, obviously the area, I mean, you know, I think you're just kind of 10, 20 years ahead of everyone else. You're totally right. But I just think the technology is not there yet. So we'll see how that goes. Yeah.
All right. Thank you.
Any more questions? All right. Okay. All right.
All right. Yeah.
Thank you.
No, thank you very much. I really appreciate it.
Thank you.